Welcome back for the last part of this lecture. Um, and this last part is about linkage disequilibrium. And so, again, this is a statistical concept, but it is heavily rooted in a biological phenomenon called meiosis. And so, in sexual reproduction, gametes are produced during a specialized cell division that is called meiosis. And that meiosis has different multiple phases. You may probably recall some of them from some of your high school lectures. We have the prophase, the meiosis one, and meiosis two. But in a nutshell, what happens during those phases is that genetic information get exchanged between homologous chromosomes, and that exchange is referred to as recombination. So what I'm showing on the right-hand side of the slide is a very quick schematic on my notebook of how that looks like. So let's say we have the two homologous chromosome, let's say chromosome 1 in mom, and we have the two homologous chromosomes in dad. So none of those full entire chromosomes will be passed on to the next generation, to the kids, but we'll have some recombined version of that. So we can see here that we have the maternal chromosome and the two of the maternal chromosomes here. So we have this version here in this version. And during meiosis, you have a recombination of those chromosomes that's going to happen, and that will produce this recombined maternal chromosome. And the same will happen in fathers. We have this recombination happening, which here you can notice it happened at a different part of the chromosome. And that will produce the recombined chromosomes. And those two recombined chromosomes, the maternal one and the paternal one, are the ones that are going to be transmitted to the kids. Um, and so, what is important to notice in that process is that recombination happens such that blocks of chromosomes are transmitted together, right? So, if you look at, for example, the maternal chromosome here, we have this part and this part, those two crosses. They also found in the recombined chromosomes, whereas some of the circles observed here in the maternal chromosome are also found in the kids, right? And so, which means that blocks of DNA that are close together on the chromosomes tend to also be transmitted together, and that at the population level, generate a correlation structure that is going to be called linkage disequilibrium. So now I'm going to introduce the concept of haplotype, which is going to be key for us to understand or also manipulate what that linkage disequilibrium concept is. And so we talked about genotypes and before, but haplotypes now are about groups of variants, but they're no longer defined by variants that are present in mom and dad, just like the genotypes. But now haplotypes will be observed within one of the parents. Let's say what I'm showing here, we have the maternal chromosome here, and we have now looking at two positions on that maternal chromosome. And just in front of it, we have the paternal chromosome. So here, meaning that this is the chromosome bit that I've inherited from mom, and this is the chromosome bit that I've inherited from dad. And so we're looking at the same positions, and we have the combination. So previously, we defined the genotypes as a combination of those two things. And let me highlight this with a laser pointer here. So those will be our genotypes. So at this position, we have the genotypes, and at the other position, we have the genotypes. But the haplotype is different because now we are looking across positions in either the maternal or the paternal chromosome. And so here we can define the haplotype as a group of variants on those two variants. And those two group of two variants can also be in multiple states, right? Here we have one state that is observed, which is the capital 1 and capital 2. That defines one potential allele for that haplotype. But of the haplotype defined by those two variants will have a different state, a different allele in the paternal state. Here it's going to be capital A1, capital A2. Two things to say about this. The first thing is that haplotypes are here to represent this notion that groups of variants that are close together tend to be correlated and transmitted together. But there is also this important notion that haplotypes are not defined in somewhat ad hoc way, right? So we know, well, I guess the boundaries are a bit arbitrary, right? So you can define a haplotype between this position and that position of the genome, and that is your definition of the haplotype, right? And there's nothing wrong if somebody says, oh, actually, I'm interested in a different haplotype that is defined by that other position and this other position as well, right? So there is a notion where haplotypes are also slightly arbitrary in their definition, but as long as you're clear in sort of specifying what are the genomic positions you're looking at and what are the possible states of haplotypes that exist, you can manipulate those concepts very clearly and rigorously. But for simplicity, in the remaining part of this lecture, we're going to only look at haplotypes that are defined by combinations of two variants. So let's now introduce a statistical definition of a linkage disequilibrium. So now we're going to consider two loci. We have locus J and locus K, and assume that at this loci, we have two possible states. We have the small a and the capital A state. So we have the small a J and the capital AJ possible state alleles at that locus J. And we have the small a K and the capital AK states or alleles that are that exist at locus K, right? And so linkage disequilibrium is a statistic that will apply between two alleles or between alleles at different loci. And for example, we will define the linkage disequilibrium between allele A small aj and capital AK at locus J versus locus K. And the way we're going to define it, and you will see it's actually quite intuitive. It is defined as a deviation, right? So it is the frequency of that particular haplotype, saying if I'm looking across locus J and locus K, how many times in the population have I seen small aj and next to capital AK, right? And I can quantify that in the population, have a frequency. But then I'm going to contrast this with the frequency of that particular combination if transmission of allele J and allele small aj and allele capital AK were completely random. If there is no correlation at all, you would expect that transmission to be random, such that the frequency of that combination, if transmission was completely random, would simply be the product of those frequencies, of the frequency of the one allele times the frequency of the other allele, right? So we're looking again at the proportion of individual in the population with both allele small aj and AK in that particular haplotype, minus the product of those frequencies independently, which will be what you would expect if there is no correlation essentially between those alleles. And so what I'm showing here is a way to represent this. So let's start with ignoring those DJK, which is the linkage disequilibrium statistic. So if you ignore the DJKs, you only look at a table with PJ, PKs, and all that. This is essentially the expected frequencies you would have if you just take completely independent loci, right? The frequency, the probability of observing together allele J and allele K, if they are independent, well, it's just a product PJ times PK. And the same thing if we want to observe capital AJ with small ak, well, what is the frequency of capital AJ is 1 minus PJ times the frequency of AK, which is PK, right? And so again, now we're bringing back the linkage disequilibrium into this. This is essentially saying that linkage disequilibrium will add to that probability of observing AJ and AK, but it had in some way that is actually structured, right? So here you see that addition on the diagonal is plus djk, and on the off diagonal is minus ejk. And one of the reasons why that structure emerges is because we know that the marginals of that table are constrained. When we have to sum all the allele frequencies, they have to be pk, or the frequency of pk, and the frequencies of each of the sum of the columns and rows are actually constrained, and that also create that particular constraint. So what can we say about the sign of that linkage disequilibrium statistic? So when dj k is positive, so we say it's in positive linkage disequilibrium. So those two alleles are in positive disequilibrium, linkage disequilibrium. If they are more often uh, transmitted together that you would expect by chance, right? But that DJK can also be negative, meaning that in this context, those two alleles will be less often observed together than what you would expect by chance. So that was the sort of the definition of linkage disequilibrium that is um, based on the allele frequencies, but also it's not the test statistic that is classically used. Usually we work with a normalized version of that statistic. We work with a statistic called the D prime, which is again a normalized version of D. So it's D prime of JK. Here is this essentially DJK divided by D max. And here I'm showing that D max here. The bottom line is you can think about that normalization as a way to ensure that we get a statistic that is still signed. It can be positive or negative, but in absolute value is between -1, is lower than 1 in absolute value, meaning that the perfect linkage disequilibrium will be when D is either -1 or 1. The other way to normalize this is to normalize relative to the allele frequencies, and that's typically referred to as the R square, which again is our D statistic square, but normalized by those product of allele frequencies. And that normalization achieves two things. First, we're losing the sign. So now that statistic doesn't know necessarily if we have in positive or negative linkage disequilibrium, at least between those two alleles. We just know that we have a certain strength of linkage disequilibrium. But it is normalized such that now it lives between 0 and 1, such that the maximum value you can observe here is 1, meaning, and when you have 1, it means that your alleles are perfectly correlated, that whenever you see a particular allele at that particular position, you are certain to see the other allele at that other position. And so just quickly how you can calculate this. So if you have allele counts, you can go through the sums and calculate the frequency of of each allele. You can calculate the frequency of the combination. For example, of AGAK, we have that frequency, which is 1,000 people with the AG and AK calculation out of 5,000 individuals, and that gives us a frequency of 0.2. And if you calculate the linkage disequilibrium here, we're getting 0.2 minus 0.25, which is a negative gametic phase disequilibrium, sorry, negative linkage disequilibrium. And then you can look at the normalized version, and here I'm mostly showing the normalization by the R square. And we can verify that the R square is the same for all combinations of alleles. If you were to apply the same strategy, you get the same notion. And that R square in this context is going to be 0.04. There is another way, if you have it from the frequencies, you can also calculate the linkage disequilibrium just as a determinant of that 2 by 2 matrix. So you multiply the diagonals minus the product of the anti-diagonals, and that will give you the linkage disequilibrium. So now I would like to end with two important aspects about changes of linkage disequilibrium. The first one is the change of linkage disequilibrium over time, and the last part will be about changing of linkage disequilibrium between populations. So let's consider two loci. So we have a locus at a particular position, another position 1, and a locus at another position 2. And we have two alleles where we have alleles 1 and position 1 and alleles A2 at position 2. So we're going to denote Rn as the frequency of that particular haplotypic allele, A1, A2, in the current generation. And we're going to denote C as the probability of recombination between locus 1 and locus 2, right? So if there is recombination between, it means that those two alleles that were together in that population, or in the current generation, or in the current parents, if there is recombination, it means that that association is going to be broken, right? Again, they're traveling together, and suddenly this one will have a different, I guess, this one will have a different allele coming with it, right? So, how can we now write this? Well, we can use that notation to try to track how the frequency of that particular allele will change over time. So what would be the frequency of that haplotypic alleles, the fact of seeing A1 and A2 together in the next generation, in generation T plus T, T plus 1? Well, it's going to be here, if we apply the total probability theorem, it's going to be the probability of having that in the current generation, if times the probability of no recombination. So if there is no recombination, it means that frequency is going to be unchanged. So whatever we had in the previous generation is going to be maintained. But that only happens if there is no recombination, and the absence of recombination happens with probability 1 minus C. But if there is recombination, meaning that their association is broken, then with that probability of recombination C, now the frequency that we observe of A1 and A2, well, now they're broken, they are independent, and now we're back to the product PA1 and PE2. And so if we do that, we can sort of rewrite that a little bit differently in terms of linkage disequilibrium, just recognizing that we've defined our linkage disequilibrium test statistic essentially as the deviation of that probability of co-transmission minus the product. And that could essentially lead us to rewrite things such that the linkage disequilibrium between allele 1 and allele 2 in the next generation is equals the linkage disequilibrium in the current generation times 1 minus the probability of recombination. So where this is taking us, it takes us that if you generalize this, it means that there is a decay that you expect over time, that over time, position, linkage disequilibrium between two positions is essentially shrunk a little bit by 1 minus the probability of recombination. So if the probability of recombination is very high, well, that decreases very sharply. If it is low, it will decrease by a small amount, but it will decrease essentially exponentially with that shape. So linkage disequilibrium at time t is a linkage disequilibrium time 0 times 1 minus c, probability of recombination to a power t. And if you were to represent that in R, that will give you a function like this. So if you think about a recombination of 1%, you start with two positions that had a linkage disequilibrium of 1. After more than 100 generation, that linkage disequilibrium will drop all the way to 2.4, right? Even lower than that. So the conclusion here is that linkage disequilibrium is not fixed. It does change over time, and under somewhat simplified assumptions here, we can predict how much it will change, it will decay over time. But there is another source, an important source of variation of linkage disequilibrium that is related to, I guess it differs between populations. And that's partly related to demographic history, the fact that modern populations have been founded by different numbers of people. So the amount of genetic diversity that different populations, contemporary populations, sort of started from is a bit different, and that will have an impact on linkage disequilibrium. But also, we could also have other pressure, environmental pressures that will lead to selection, natural selection, to have change the certain combination of alleles, the frequency of certain combination of alleles, which is essentially changing linkage disequilibrium. So I think we could have an entire lecture on that particular part. I just wanted here to emphasize the empirical data around that difference. And that was, for example, shown in this 2015 paper showing data from the 1000 Genome Project, which I've mentioned before. And here on the x-axis, we have the distance between markers in kilobases, so from 0 to 100 kilobases, so 100,000 base pairs, essentially for the maximum value. And the y-axis shows the average R square, which I introduced as an LD linkage disequilibrium statistic. And so there are a couple of things to observe. One is that linkage disequilibrium does vary with distance. So the further apart the variants are, the lower the LD. And this is a property that is also important to keep in mind. It will influence many of the applications that you're going to hear about in the following part of the workshop. But also, it shows that there are differences between populations. And here, just looking at different populations across the world. And in particular, we see that all the African ancestry populations are somewhat detached and at the bottom pretty much showing that they have lower linkage disequilibrium, and that is related to this demographic history that was mentioning earlier about the sort of effective population size or the size of the founders that have led to those contemporary populations. So that's also something to keep in mind. All right, so I'm just now going to summarize, and that will be the end of this video. So what we covered here, we covered in this lecture that the concept of Hardy-Weinberg equilibrium. We talked about some of the drivers of changes in gene types and allele frequencies, in particular, genetic drift, sorry. We also talked about measures of genetic distance between populations with FST statistic, and finally introduced linkage disequilibrium, which is a measure of genetic similarities between parts of the genomes that can be further apart, and also shown that linkage disequilibrium, sort of the statistic association between the frequency of alleles at different parts of the genome does vary between populations and as a function of the distance and over time. I guess with that, I will thank you for watching this video again, and I can't wait to meet you online and have a chat about the practical and hear your questions. So thank you very much for your attention, and this is just another final slide that we're going to keep in mind to play with during the practical. Thanks.